Tell me some things you notice about it. So purines always pair with pyrimidines. What else?
How do they fit so easy if it's so hard to fit on here?
Remember, in 3 dimensions is how they're aligning. So you guys, to get them to fit quite as easily as they do in reality, would have had to trim pretty closely to your pieces, to your like paper pieces. Those of you who trimmed closer rather than like making bubbles, they fit a little bit more neatly. What other observations do we have? So the sugars and the phosphates are linked. They make like a ribbon down each side, right? So sugar and phosphates link together.
This is what you want on your metals.
What else do you notice?
Like one side's upside down.
One side is upside down.
Is that so it can get the mirror image?
That's what makes it anti-parallel, right? In order to make the bases fit together like a puzzle, the way they're supposed to be complementary, one side has to go in the opposite direction of the other. So it's anti-parallel. And it's easier to see that they're upside down on your models because you actually have letters that are upside down looking at it, right? In my picture here, they oriented it so all the letters are right side up, and it's less obvious that they're upside down until you take a look at where the oxygens are located, the way they're pointing on the deoxyribose. What else do you notice? For every adenine, there's a what? And for every guanine, there's a cytosine. So we actually have a name for that particular rule, that particular trend where A equals T and C equals G. We call that Chargaff's rule. Named for the scientist who said that the percentage of A will always equal the percentage of T, and the percentage of C will always equal the percentage of G. How many rings across is your molecule? So you told me that purines always pair with pyrimidines. How many nitrogen-based rings? 3. So it's always 3 rings wide. And I was out there amongst you guys when some of you mistaped. And then you looked at me like, oh wait, that's not right. That doesn't go together. It's too wide. If you had 2 purines together, it was 4 rings wide, right? And would your sugars and phosphates line up to make nice even lines if you did that?
No.
No, so it's a base pairing mismatch. This particular diagram, we'll make it a bit bigger so we can look at it more closely, shows us our, we'll just move Chargaff's rule over here, shows us our anti-parallel base pairing. And it shows us something special that happens with red lines. In between the nitrogen bases, those red lines right here represent hydrogen bonds. So as I was out there helping you guys pair, you'll notice that I rotated each of your bases until they lined up specifically. The reason that they have to pair complementary is to make sure that the correct atoms are next to each other to create those hydrogen bonds. There's always going to be 3 hydrogen bonds that occur between guanine and cytosine, and 2 that occur between adenine and thymine. That only works if it's anti-parallel. If they weren't anti-parallel, this couldn't happen. Wouldn't work. You guys see how that works? We're good with that? This anti-parallel thing's not quite as bad as the first time we saw it, right? You're like, no, okay, I get it. I see why it has to be the way it has to be. Ah! Taking a look at one of your colleagues' models then.
That looks so much better.
We can annotate it. Here's my sugar phosphate backbone. Oh, I'm going to choose a fancier color. I want a fancy color. That's a fancy color. Here's my sugar phosphate backbone. Here's the other anti-parallel sugar phosphate backbone. And then I'm just going to draw a beautiful green line between my nitrogen bases. So I've drawn in some hydrogen bonds. That looks like all of the cartoons you've ever seen now, right? Where you have rungs of a ladder going up and the sides of the ladder. The only thing that happens now is we take this thing and it gets twisted. Now, something tells me, since we struggle with tapes, that origami is out of the question. So we're gonna not get too ambitious. Okay, we're honors biology, not honors paper folding, so we're gonna not push too hard. That's how all of the DNA you have fits in every single cell. In that nice little pattern that's anti-parallel. Every cell contains your whole genome. Genome's a crazy fancy word, right? It's a crazy fancy word that simply means every nucleotide an organism has. If I could spell. Spelling today, apparently. We're on the spelling struggle bus. One more thing that's kind of important about that genome thing. It's every nucleotide the organism has in order. So you guys asked me when I told you to extend your chain, you're like, does it matter what goes next? Like I have this A, T, C, G thing. Do I have to go like A next? Does it have to repeat the same order as above? And I was like, no, just whatever order you want. Whatever order you please. And the reason that's true is because what makes every organism different is the different order that those A, T, C's, and G's occur in. That's why we're different. That's why we look different. That's why we don't look like elephants and we don't look like reptiles. Because our ATCs and G's and their ATCs and G's are all in different orders. So, there's that. That's, we call it the genome. That structure. All 6 billion bases, 3 billion base pairs fit in every single cell you have. Now here's the key. Remember that whole mitosis thing from last chapter? Remember S phase of interphase?
Yeah.
All 6 billion nucleotides have to be copied in the exact order they occur in every time a cell divides. That's crazy, right? That's what's going on during synthesis. And it has to do it in order with no mistakes. How's it going to do that? Photocopy? Listen, making photocopies is actually a lot more difficult than you think. This morning, I was on the photocopy struggle bus. Photocopier kept beeping at me, telling me that it wasn't oriented in the right way, and it didn't have the right size paper, and I kept violently hitting the blue button to try and make it go. It's not as easy as you think. They don't teach you that in teacher school. They give you all kinds of other classes. Nobody tells you how to use a photocopier. Yeah?
They teach you guys how to be teachers, not just information.
Yes. The information we had to teach, we had to know before they taught us how to be teachers.
Oh, so you don't have to, you can have to show you the information.
Oh no, you have to take these really big, long, hard tests.
So even when you're a teacher, you take more tests.
The only way they give you a certificate is if you pass your tests in your content areas. And the only way you can get a job is if you have a certificate. Mm-hmm. So they make you keep taking classes to remain highly qualified.
When you said that the sugars and the phosphates link together,
Uh-huh.
the phosphate is the little,
The little P with 4 oxygens. Yep,
and then the sugar is the deoxy.
exactly. So somehow the cell had to devise a way to make a perfect copy of this thing during the S phase of interphase before we can enter mitosis. That process is called DNA synthesis, also known as replication. The process we now know as DNA synthesis or DNA replication is semi-conservative. What does semi mean? Partially. And if you are trying to conserve water or conserve resources, what are you trying to do? Save them, right? You're trying not to waste them. So half saved. That's what this model is. And I'll show you guys a video of the experiment that we learned this from. It's called the Meselson-Stahl experiment, named for the scientist who did the experiment. You'll notice when we watch the video, it was done in bacteria. This is not something you're going to do in a people. And the reason why will become abundantly obvious when I show you the video. So, the Meselson-Stahl experiment. There is indeed a difference between the way Prokes and Ukes do this. Replication is going to start at the beginning. It's a good place to start, right? We start replication at the origin of replication, origin meaning beginning. And it's going to move in two different directions from that origin. So we have our original strand. I apologize for some reason. When you see DNA replication, when you see DNA itself, it's pictured up and down. But when you see replication, we take the molecule, we lay it on its side. So if you're having a hard time seeing it, here's our sugar phosphate backbone on our sides. There's the sugar phosphate backbone. There's the sugar phosphate backbone. Here are nitrogen bases. And somewhere in there is an origin, and that's where the molecule is going to open for copying, at the origin of replication. Now remember I said this is different between Prokes and Ukes. Prokaryotes have one origin of replication. They have one origin. It's going to start at that origin, and then it's going to move in both directions along the origin, making what we call a replication bubble. So we call this big thing right here a replication bubble. And replication is going to move right, and then left. We usually call it downstream and upstream. In a eukaryote, many origins of replication. That's how it's faster, because it starts at different spots, and it stops when those spots meet. So we can speed it up a little bit. Part of the reason that's true is because in bacteria, their chromosomes are circular. They're round. In eukaryotes, the chromosomes are linear. So we have to have it in multiple spots to make sure we get the whole linear strand. The circle, it's going to be easy because it's just going to go in a circle until it meets itself on the other side. This one? That's the original strand of DNA. So whatever exists in the cell. And then we have to open it to make a copy of it. These are both in eukaryotes. Both Prokes and Ukes. This will happen.
So like in eukaryote with that, like in strand one just open up here and then open up here and then open up the wild.
So the difference is in a Proke, this would be their DNA in a big circle. And there's only going to be one origin, and it's going to open and move in both directions until it meets itself on the other side. Here, in a eukaryote, they're linear, they're straight lines, and it's going to open in multiple spots along that big line. So it can't ever meet itself at the beginning again because it's not a circle. It's a straight line. You guys get that?
What is the first, what kind of molecule?
There are a whole bunch of aces here. Anytime we see the word ace, you guys know what it means when you see ose, right? It means sugar at the end of a word. Ose means sugar. Anytime you see ace at the end of the word, it means enzyme. So we are going to be introduced to all kinds of new enzymes this chapter, next chapter, and the one after that. The major players here are some crazy enzymes with some crazy names and some less than crazy names. Topoisomerase, yeah, that's a fun one. Helicase, primase, and DNA polymerase. So those are all of our ACEs. At this point, remember what an enzyme is. So an enzyme is a chemical that speeds up reaction without being used or without being part of the product. So it's basically like a chemical helper. All of these things are chemical helpers to assist in our 6 billion base pair photocopying. So they're all pretty important. I'm going to kind of talk you through replication first, and then we're going to see what replication looks like in motion. I promise it will make a lot more sense in motion. Sydney?
What's a chemical called that slows down like that?
An inhibitor.
What is the thing after speeds?
Reaction. In chemistry, we're very lazy. That's why we rely on so many formulas and abbreviations. And RXN is the standard abbreviation for reaction.
So you said an inhibitor slows down there?
Mhm.
Alright.
So all of these things, they're designed to make this process faster. And we said it has to be pretty fast because we're trying to fit 6 billion during that S phase of interphase, a copying of 6 billion things during that little wedge of the cell cycle. Has to be fast, has to be accurate. These players are actually all listed in order. This is 1, 2, 3, and 4. And I'm going to have you think about when you were a kid and you sat on the swings on the playground and you spun in a circle to see the chain of the, does it make you sick?
No.
To see the chain of the swing curl up and then get shorter and shorter and shorter, right?
Can't touch the ground.
So that's what DNA when it's all curled up is like, right? It's all like bundled and almost knotted. And to start replication, we have to go in there and cut the hydrogen bonds that occur between nitrogen bases and peel open the 2 strands of DNA that are next to each other. Think about just the physical ability to do that. When you're on that tightened, coiled up swing, can you take the 2 chains of the swing and pull them apart?
No.
Can't really do it. So one of these chemicals, its sole function is to release the tension so it can be opened. So that's its job, is just to physically release any of the, what they call it, torsional stress. The stress of being twisted. That's what the topoisomerase does. It's just to relieve any of the stress of a curled up double helix. And helicase has another important job to start this whole thing too. We said yesterday that those hydrogen bonds naturally happen when those 2 complementary base pairs line up next to each other. Think of it like magnets. When you put 2 magnets that are attracted to each other near each other, if they're close enough, they just stick together, right? Those nitrogen bases will do the same thing. If they're close enough, they stick right together. You can't prevent it. You have to apply some kind of energy to pull them apart, right? Helicase's job is to come in and just pull them apart.
You said nitrogen bonds?
Hydrogen bonds between the nitrogen bases.
Wait, do they both hold apart?
This one releases the tension, so this one can come in and pull it apart. At this point, when we talk about replication, we usually refer to DNA as a zipper. And we say that helicase unzips. So it's going to open it like a zipper. What I think is probably, when we look at the zipper thing, is probably a better way to look at it is, you know those zippers, those coats you have that have 2 zippers? Like, you can unzip it one way and the other way.
Those are, wait, what?
Yeah, I'm
North
good.
Face
Yeah.
coats do that. Like ski coats do that.
Yeah.
You're talking about the one with like the under zipper. That looks like, oh no.
No, it's got the only thing I can think of that I own that has it on it.
Because it has two.
Yeah, you can use a backpack. So it's got 2 zippers.
Oh, no.
So like your backpack with 2 zippers, picture DNA like that zipped zipper. And the point of origin, the origin of replication, is when you have it all zipped up and both zippers are right next to each other. And in order to start replication, they have to unzip. They're going to unzip in both directions, making one bubble in the middle. We call that a replication bubble. So that's what happens. Helicase comes in, acts like those zipper pulls, and just pulls them apart and opens them up. Topoisomerase has already come through and released the tension so it can get unzipped. And then helicase comes through and unzips it. That's how this thing starts.
That, like tofu.
Isomerase?
Yeah. It's like the annoying kid that was like pull on your backpack and try to loosen it.
Mm-hmm.
I hate those. I was one.
The fact that you said that they were the annoying kid and then you labeled yourself as one of those. That's good.
They annoy me like that. So does the helicase pull away like the staircase, or does it?
It cuts the rungs of the staircase in half.
Oh, so like the picture from yesterday with the like bubble.
Mhm.
All right.
So we said that topoisomerase releases the stress, the tension. Yep.
So, in the 2 pictures you gave us yesterday, so, like the first one where it's just straight.
That's just before replication has begun.
Okay, so, like that's when like the tension is
Uh-huh.
released, and then it like unzips and makes the bubble.
Exactly.
All right.
So then helicase comes through and unzips. And the problem here is that, remember we said that if those nitrogen bases are close enough together, they automatically reform. That bond just like forms. We can't stop it. Kind of like magnets, if you pulled them apart, if you left them close together, they're going to reattach. But if you put something in between them, they're not going to, because you have a physical barrier. So here we have these things called single-stranded binding proteins. And those single-stranded binding proteins prevent the helix from re-zipping. It prevents the helix from re-zipping. Once we have that open bubble, the third enzyme is free to come in. And the third enzyme is called primase. Primase attaches an RNA primer. Have you heard the word primer before?
Yeah.
What does it refer to?
Like, I hear when like people are painting.
When people are painting.
Isn't it like a base coat?
So if you have fresh drywall on the walls, like fresh sheetrock, never been painted before, it soaks up paint. It's gonna like soak it up like a sponge. And good paint is really expensive. The stuff that you see on your walls that has color in it, super expensive. So you don't want to have that drywall suck up all the paint, and then you have to keep putting like coat after coat after coat on. So you put on a base coat called a primer. It gives something for your good paint to attach to. That way you're not wasting the good paint. You'll never see the primer again. It's under there. You use primer especially if you're going to take a wall that was nice bright white and paint it like a dark rich color. You use a tinted primer to help that color adhere. When we start replicating these strands, the new nucleotides in the new strand have to have something to attach to. So they attach to a primer. But the primer doesn't come out of nowhere. It doesn't just magically appear because it's needed. It's attached by this enzyme, primase. Much like anything else in biology, when we give it a name, we try and name it in a way that indicates its function. Helicase, ACE is an enzyme that unwinds the helix. Primase, ACE is an enzyme that attaches a primer. Don't ask about topoisomerase because I don't know who invented that one. They do everything else all nice and obvious, right? And then
Then they gotta have the one.
they throw in that first one. They're like, just in case you thought these were too simple. Biology. After that primase attaches an RNA primer, our last enzyme comes in, and its name is DNA polymerase. ACE is an enzyme, and it makes the DNA polymer. That's where it gets its name. So it's going to attach complementary DNA nucleotides to the primer. Now, I will tell you, DNA polymerase has a teeny tiny, it's not a flaw, it's more of a quirk. What direction do you read when you're reading in English? You read left to right, right? You know there's some languages that they don't? They read in the opposite direction?
Yeah.
But what would happen if you tried to read an English sentence?
It would look. It would make sense.
Yeah, you're like, you can't start on the wrong half of the page and read that way. DNA polymerase can only read in one direction on the strand. So the template strand, remember how we labeled them 5 to 3 and 3 to 5? So we have our template. And it's opened because helicase came through and cut it open. Primase came in and attached a primer of complementary RNA bases. Now DNA polymerase is going to come in, and it's going to try and attach bases. But it can only go in one direction on the strand. It's only going to read the template from, let's see, this is the part that always confuses you guys. How do I say it? I got to choose my words wisely. Maybe I'll just show you. Here's our friend DNA polymerase. It's going to be attaching complementary base pairs. Here's our template strand. It's moving this way, growing the new strand. So it can only read the template from 3 to 5, which means it builds the new strand from 5 to 3. Because the strand directly across from it's always going to be anti-parallel, right? Yeah. DNA polymerase can only move on the template from 3 to 5. Now, right now, you're like, this doesn't seem very complicated because helicase is just going to keep unzipping going this way, right? So it's going to keep unzipping, and DNA polymerase is just going to keep adding until it reaches the next origin of replication, and one had already started there, and then it's done, right? That works brilliantly on the template strand that is 3 to 5. But what's the template that was across from it? DNA polymerase can't just move that way, can it? So it's got to go the other way, right? Gets a little bit tricky then. That's not the tricky part.
That.
It's okay. It's okay. We're here for another 31 minutes. I can't possibly talk about this for another 31 minutes. So my template strand here is blue. Agreed? Here's the nitrogen bases. Green is our friend helicase. No, I'll leave that one. Pink is going to be, well, we'll label it 3' on my template to 5' on my template. So the strand across from it is what? He was right. You're 5 to 3. RNA, the primase has attached our complementary RNA primer right here. And then DNA polymerase came in. Ooh, let's make that white. DNA polymerase has attached complementary base pairs. And helicase is going to keep moving this direction, unzipping, right? And so this DNA polymerase can keep following. It's just going to keep following as it keeps going, adding more complementary bases. Life is good. See how that works? That one is fast. That top strand is going to be quite snappy because it's just going to keep reading. It's like if somebody kept typing a word and you kept reading as they were typing, you'd keep up with them, right? So we call that top strand, because it's so fast, we call it the leading strand because it leads the way. It's fast and it's continuous. Here's the problem with the bottom strand. Helicase is going to keep moving this way. DNA polymerase can't read that way. It'd be reading the sentence backwards. So instead, primase comes in and attaches an RNA primer right here. Then a different DNA polymerase can attach complementary base pairs moving in this direction. So it moves reading the template strand from 3 to 5 that way. You see how that makes sense? You're okay with that, right? You're like, all right, that's fine. It's reading from 3 to 5. It just attached the primer here and then works backwards. Mm-hmm.
How can it unzip itself if one is going?
So, but doesn't that mean that here, as this helicase moves that way, you're going to have a bunch of unreplicated DNA right there? That's precisely the problem. So on that strand, you're going to have another primer come in. As it continues unzipping, another primer is going to come in and attach and work its way back to where the first primase was. And so it has to do it in tiny chunks. And as soon as it gets back to where it started, it releases and it's done, and then it moves on to where it's then unrezipped again and has to work. So it works in pieces, tiny fragments. Working in fragments like this, super slow. Super slow. It lags behind in time, so we call it the lagging strand. So we have our leading strand on top that's continuous, our lagging strand on the bottom that's discontinuous because it's done in pieces. And those little pieces have kind of a weird name. On the lagging strand, when it's done in pieces, we call it Okazaki fragments. That name comes from the scientist who discovered the fact that this is in fact discontinuous. So they named it for him. They are Okazaki fragments. This process is happening in a eukaryote multiple places along the chromosome. So in all of the DNA, happens in lots and lots of different places. And the reason that happens in many places is because eukaryotes have so much DNA, you have to start replication at a whole bunch of different places, because that way it's faster when you put the whole thing together. I was going to compare it to something and then realized for you guys, completely irrelevant comparison. Yeah?
So are like the primer and the lagging strand, the primer and the leading strand, are they going opposite ways?
So the topoisomerase actually fits around and relieves that twisty tension and continues to work its way in both directions. Here, helicase, rather than looking like a blob that attaches, looks like a jabby thing that can cut. Different depictions, I don't know.
So it doesn't show the topoisomerase.
In the previous drawing, no, or in the previous animation, there was no topoisomerase.
Okay.
A lot of textbooks are going to oversimplify this process. So, for example, one of the textbooks that we use, well, you guys have never seen it, it's this textbook doesn't even talk about leading versus lagging or Okazaki fragments. It just like talks about one strand and that's it. So that's why you don't get taught out of this textbook. They don't learn all of it. They learn the simplified version. So that's why you didn't see topoisomerase in that animation, because even that one simplifies it. Truthfully, what you're actually looking at is simplified in and of itself, because there's actually 3 DNA polymerases, DNA polymerase 1, 2, and 3. And they all have different jobs, but I'll let you worry about that when you get to college. Yeah. Here's just one.
What would happen if the Okazaki fragment weren't present?
Then that second strand would never get made. So we would only have like 1.5 DNAs. You'd have the leading strand that was made and replicated, but the bottom strand would be half complete.
So is it just that?
Mm-hmm. So this one is from your textbook, and it shows you your leading strand, 3 to 5 prime, your lagging strand, 5 to 3 prime, and it shows you the Okazaki fragments being made in their little pieces on the template, and then the next Okazaki starts here, and then you keep going. Your textbook does give you polymerase 1, 3, all the different ones. Not too worried about those. But you know how I said it's DNA polymerase that goes back through and removes the primer? It is DNA polymerase, it's just the other DNA polymerase. I don't care if you know the difference, I just want you to know that it's DNA polymerase and DNA polymerase. And they all serve the same function. Isn't that pretty? That's not so bad now, right?
Looks like a little racetrack.
It does look like a little racetrack.
So on the lagging strand, there's more DNA polymerase.
Yes. Well, more in quantity, yes. Like there has to be more of them to keep doing it. Because in reality, these don't stretch out into a straight line. They're actually like knotted and curled up. I'll show you. I've actually only drawn you half a picture. I've drawn you this side of the picture, right? The exact same thing is happening on the other side. So there's another helicase moving in the opposite direction. So that polymerase can keep moving in the opposite direction. But if you're thinking,